Glycogenolysis. What is glycogenolysis? Glycogenolysis is the chemical breakdown of stored glycogen into glucose to provide immediate energy for the body. This maintains blood sugar levels of the body. Where does it occur in the body? This metabolic pathway occurs primarily in the cytosol of liver and skeletal muscle cells in response to hormonal or neural triggers. While the liver exports this glucose into the blood to support the entire body, muscles use it locally to power contractions. So a quick breakdown of glycogenolysis. It is derived from two words. Glycogeno involves glycogen, the stored sugar, and lysis means splitting or breaking apart. Where it happens? It happens primarily in the liver and skeletal muscles. Why it happens? To raise low blood sugar levels or to power active muscles. Which hormones control glycogenolysis process? Three main hormones control glycogenolysis by signaling whether the body needs to release the stored sugar or whether the body should save it. Hormones that turn it on, causing activation of glycogenolysis. During fasting, low blood sugar, or a fight or flight response, stress. The pancreas releases glucagon, and the adrenal glands release epinephrine. These hormones bind to cell receptors, triggering a cyclic AMP cascade. This cascade activates protein kinase A, which phosphorylates and activates glycogen phosphorylase. Hormones that turn it off, causing inhibition of glycogenolysis. After eating, high blood glucose levels trigger insulin release. Insulin activates phosphatases that dephosphorylate glycogen phosphorylase, rendering it inactive and turning off glycogen breakdown. Now let's look at the step-by-step cascade of glycogenolysis. Here is how the enzymes physically break down the glycogen polymer in glycogenolysis. Step 1. Cleaving the main chains, the linear bonds. The enzyme used is glycogen phosphorylase. Its active form is phosphorylase A action. This enzyme is the main target of the hormonal cascade. Once activated by phosphorylation, it attacks the linear alpha 1 to 4 glycosidic bonds at the outer ends of the glycogen molecule. Mechanism. It uses an inorganic phosphate to split the bond, releasing glucose 1-phosphate. It repeats this step sequentially, shortening the chain 1 glucose unit at a time. Step 2. Handling the branches, the debranching process. Glycogen phosphorylase cannot get close to branch points. It stops exactly for glucose residues away from an alpha-1,6 branch link. A single bifunctional enzyme takes over. This enzyme is glycogen debranching enzyme that possesses two distinct catalytic activities. Step 3. Rearranging the sugar. The enzyme used is phosphoglucomutase. Action. The massive pool of glucose 1-phosphate produced in step 1 cannot enter metabolic pathways in its current form. This enzyme shifts the phosphate group from the carbon 1 position to the carbon 6 position. Thus the product is glucose 6-phosphate. Step 4. Creating free blood glucose, liver only. Here the enzyme used is glucose 6-phosphatase. Action. The muscles lack this enzyme, so their glucose 6-phosphate is forced into glycolysis for local energy. However, the liver uses this enzyme inside its smooth endoplasmic reticulum to strip the phosphate group off glucose 6-phosphate. The product is free glucose, which can safely cross the cell membrane into the bloodstream to raise systemic blood sugar.